My name is Lee. My full name and title is Professor and Dr. Lloyd Lee Glenn. I worked at the College of Nursing and I'm trained as a neurobiologist. The video that you're about to see is on brain anatomy. It was taken during a live brain anatomy sessions with doctoral nursing students in the area of psychiatry. And it goes over all of the major anatomical structures of the brains that are important in psychology and psychiatry. It is not really designed for a neurological type of course, even though we do cover very briefly things like reflexes and go over brainstem functions. We focus primarily on the cerebral hemispheres and the limbic system. I hope you enjoy this video and find it useful. Hannah, how you doing?
Good, how are you?
Good.
Leah.
Leah, how are you? It's good to see you. Dr. Theodore Hag teaches the neuroanatomy with us. He's the chairman of the Department of Biological Sciences. Hello to everybody there in video land. Don't mean to ignore you. It's glad to have you in here joining us. There is a hemisphere, one of each hemisphere. This is the cerebellum attached to the brainstem. The blood supply is intact, so we can go over that. This is a cerebellum without the blood supply or the brainstem. There's a couple of these. Here we have a whole brain. I'm sorry about splashing.
Oh, it's okay.
That's why you need the gear on. Another hemisphere, another cerebellum. Here's a brainstem without the cerebellum attached. Boy, we got cerebella all over the place. Here's a half section of a brainstem and a cerebellum. Here's the brainstem, here's the cerebellum, so you can orient yourself to that. Alrighty. First, brain anatomy with the principles of anatomy, which apply to all brain sections and all structures. You've had a little bit of this in lecture, but you've not had it told to you in an accurate, compact way. The brain is most easily understood if you understand how it is created, embryology being sort of how it's constructed and built. We start out where the sperm hits the egg, creating an embryo. This embryo then starts to divide by mitosis into more cells and more cells and more cells, just splitting in half and splitting in half and splitting in half. As it gets to a certain size, it starts to take on this form. Each of these dots represent a single cell from the primordial embryo. It forms a gut, and this will eventually become the gastrointestinal system, and a skin. This eventually will form the skin, the ectoderm. It'll produce keratin, which is the plastic stuff in our skin that keeps water in and keeps our skin flexible. And then up here, it has these two folds that come up, called the neural folds. The neural folds meet up here, leaving a tube in the center, form a complete circle. That tube eventually becomes the ventricles of the brain and the central canal inside of the spinal cord, which is filled with cerebrospinal fluid. Cerebrospinal fluid, real quick, it's a very clear substance, hardly anything in it. It's almost like salt water with a few cells, a couple of transmitters, very low levels of transmitters or other hormones. People always wonder, well, is it used to communicate with one part of the brain with the other? The answer doesn't appear that way. The brain has the consistency of hard jello, and it's floating inside of the cerebrospinal fluid, which is basically saline. So if you have an impact on the side of the skull, that impact is transmitted to the opposite side as a wave that goes through, and the brain is not compressed. It's not compressed because liquids are incompressible. Brain's kind of like a jellyfish. You say, well, those things are hard. You have to use formaldehyde and harden the brain to get it out. If you open the skull of an unhardened brain and try to take it out, it will just mush up on you and just sort of pour out. It's just not possible to get it out. A whole bunch of cells get really tightly formed in here and here and here. And these were given the name plates because they're sort of shaped like a plate, like an oval or like a cigar or something. It's called the alar plate. Alar means roof because this is on the top here. And the basal plate. Of course, basal means base, it means bottom. So we have top and bottom. Everything on the alar plate develops into the sensory system of the brain, whether it's audio, visual, touch sensation, it's all sensory. And fibers start to grow out of here and innervate the tissues of the skin, the muscles, the bones, that provide all of the sensations. The ears, the cochlea, for incoming information. That's an arrow. Incoming information. Now, everything on the basal plate develops into the motor system. Movement projects to muscles, causing movement. They grow out down here and eventually innervate muscles that are beginning to form out here from aggregations of cells that will become muscles. Within this division, there's a separation between somatic and visceral. Visceral means related to the gut. The somatic part, on the edges, the sensory and the motor goes to the body. Soma means body, and it goes to muscles, skin, joints, skeleton has an innervation. Particularly the periosteum around the bones has a very strong, high-density sensory innervation. Blood vessels are considered visceral. They're innervated from these inner ones. So we have sensory, motor, and the viscerals over here, and the somas on the outside. So everybody got that planned? The whole brain is organized in that fashion. It twists and turns and loops, but it's always got that structure to it. Cortex is the last thing I'm going to have to show. But at a certain point during development, cortex appears. What happens is cells migrate out. They migrate out and form a layer, and they go to the outside, and they pass fibers on into the inside. The primordial neural tube has gray matter on the inside, white matter on the outside, and you're going to see that. Cortical tissue is different. It is new. It comes recent during ontogeny. It's one of the later building blocks, and it's a mushrooming out where you have gray matter on the outside and white on the inside. The word cortex is a Greek word for bark. Make a cross-section of a tree, you have a thin line of bark around it and all the white pulp in the middle. It's named after that. This is the bark, the gray matter, the cells is the bark, and the white matter is the core of the tree. Okay, we have for you the brain. The dura mater has been removed. This is the pia mater that covers the brain. The dura mater means dura is tough, as in duration, and mater is mother. So it's the tough mother, and it's sort of like the womb-like characteristic of the dura mater. And it is really tough. It is like leather. And the pia mater is much more delicate. You really can't see the brain structures until you remove the pia mater, which is a meticulous process. Here the pia is present, and here the pia is removed. All the arteries have been removed too. Isn't that just beautiful? I mean, the clarity it is. So if you find yourself with something that's got pia on it, feel free to peel the pia off or get a piece that has the pia already removed. Well, here we have the huge human cerebral hemispheres here. Enormous. We have the frontal cortex, parietal cortex, temporal cortex, occipital cortex, also called the frontal lobe, parietal lobe, temporal lobe, occipital lobe. More on that in a second. Here we have the sagittal fissure, and the huge sagittal vein runs through here. If we flip it over, we see, first of all, the second biggest structures are the cerebellums, the two halves of the cerebelli. Cerebellum means small or little cerebrum. And you can see why it got the name. It's smaller. These folds that you see are tinier. And it goes around the back of the brainstem. This is the brainstem. Below the brainstem at this point is the spinal cord. So we have got spinal cord here. And just above this is the brainstem. Spinal cord has gray matter on the inside, white matter on the outside. It has the primordial neural tube structure. Here we have the blood supply to the brain. And you can see here some structures. These are the vertebral arteries. They come from the heart, branch off of the aortic arch, run through the vertebrae, and supply the brainstem and the upper part of the spinal cord primarily through all their branchlets. This is the basilar artery. It is huge. It supplies the pons, and it goes into a structure up here called the circle of Willis. Circle of Willis is sort of like a distribution center where blood comes in from all sources and can go out through a number of directions. It's sort of like a switch where things can, so if you lose any blood flow from a clogged artery, other arteries can make that up from the circle of Willis. Brainstem. Well, if you see the brainstem here, you'll notice that's what I have here, right? Now, the brainstem has 3 levels. So it's the medulla. The medulla is distinguished because it's below this large structure here. And it has these 2 bumps on the side. These are called the olives. Why? Because they're the size and shape of an olive seed. What do? They're involved in audition. They're part of the auditory pathway to the cerebral cortex. These parts of the medulla is called the pyramids. They contain the descending tracts to the motor area of the spinal cord, which is the basal plate. It's called the corticospinal tract. It goes through the pyramids. Corticospinal tract originates in the precentral gyrus. Here I see the central gyrus, the central sulcus, so this is the precentral gyrus. Gyrus and sulcus, what are those? A gyrus is one of these convoluted structures, sort of the hills or the mountains. Kind of looks like an intestine or something. The valleys in between are called a sulcus or sulci. So it's gyrus or gyri, a sulcus or sulci. They vary from brain to brain all over the place. I can take any 2 brains and you will see a variation on that. Some brains don't have some of the gyrus and sulcuses, and some do. So they're approximate. What is not approximate is the areas of Brodmann. The areas of Brodmann shift. Sometimes they're down in the sulcus mostly, sometimes they're up on a gyrus mostly. But once you identify an area of Brodmann, it is fixed and it does what it does. The areas of Brodmann are the units of processing in the cortex. Units of processing. So when you think cortex, a lot of times we say, well, the precentral gyrus does this, the inferior occipital lobe does this. Really, that's approximate. What really matters is which area of Brodmann you're in, because now you're talking, because each area of Brodmann has a specific function and tends to move around. The trouble is, because of that, they're hard to find. You really can't find them on a living thing. You pretty much have to section them, go under a microscope. So here's the central sulcus again. Central sulcus right here. Starts on the inside and goes down. So this is the precentral gyrus. And this is the postcentral gyrus. The precentral gyrus is motor. It's Brodmann area number 4. And it gives rise to the corticospinal tract, which goes down, down, down through the midbrain, through the pons, and shows up in the medulla as the pyramids, also called the corticospinal tract. So here we have got the medulla, identifiable by the pyramids in the olive. Then we go up, the next structure is called the pons. The pons is this gigantonormous thing on the front of the brainstem. Go ahead and look at that, pass it around. Enormous thing here. Pons is Greek for bridge. Why bridge? Because it forms a bridge over the brainstem, appearing to go from the cerebellum lobe on the left side over the brainstem, the cerebellum lobe on the right side. So pons is bridge. It is huge. It's also called the pontine enlargement. And the corticospinal tracts cut right through the pons. And I'm going to pass this around in a second. And when I do, I want everybody to look to see if they can see the streaks, the white streaks of white matter coursing down through the pons to take up residence in the pyramids. At the level of the midbrain, which is here, the corticospinal tracts are this structure, known as the crus cerebri. So, same old thing, the corticospinal step is called the cerebropeduncles, or the crus cerebri here. Through here it's called the pontine striations, then down here it's the pyramids. It's all the same structure. The ancients did not know that these were all the same structure and had the same function, so they gave them different names. This is the base of the brainstem. This part in the middle here is called the tegmentum. The tegmentum. The brain indeed is gray and white. After you stain it with formaldehyde, instead you get dark yellow and light yellow are the colors that are in there. So the dark yellow is the gray matter. And you can see the pons has got a lot of gray matter in here. Those are the pontine nuclei. This structure right here is called the red nucleus. The main function of the brainstem is to control the cranial nerves. There's a cross section at the level of the medulla. Here you see the pyramids, and this is the olives with this convoluted cortical-like material in here. This is the pons. This is the base of the pons. This is the tegmentum. And you can see these striations in here, those stripes, striations, like the corpus striatum or the striatal striata means stripes is all it means. You see this is stripe. This is the pontine striations. And for the medulla, they sort of have two functions in the brainstem, because the brainstem is already specialized. Does your arm have any ears or eyes on it? Does it have a tongue? Can it taste things? No. That's all up in the cranium. That requires processing machinery of the brain. And that requires bigger nuclei, tracts. So the cranial nerves are no different than the muscles of the sensory and motor apparatus of the spine. It's just more specialized for our special senses at that level. So one thing is all of the sensory and motor nuclei. The structure of the brainstem is like this. Do you see how this structure here used to be the central canal in the spinal cord? Now it's the fourth ventricle. We'll do ventricles in just a minute. Second thing about the brainstem before we leave it. In addition to these cranial, nerve, sensory, and motor nuclei, there are two things that are very vital in the brainstem. At the level of the medulla in the pons contains the respiratory drive centers that project down the corticospinal tracts to the motor neurons in the thoracic spinal cord. And the motor neurons then go to our rib muscles and our diaphragm, giving us the respiratory pump. Air in and air out. If these nuclei are destroyed, you have death within seconds. Well, within a minute. Soon as respiration stops, oxygen runs out, your brain will probably go unconscious in 10 to 15 seconds, and then your heart will stop beating. The respiratory centers are very delicate and very easy to disrupt them. Same thing if you cut the tracks here at the level of foramen magnum, respiration is lost. The spinal cord cannot sustain the respiratory oscillations by itself. The pressure of the cerebrospinal fluid inside of the cranium is about 10 to 15 millimeters of mercury. Blood pressure is what? 120 over 80? Well, 10 is a lot lower than, say, 100 millimeters. If we average 80, 120, maybe we get 100 millimeters. What do you think happens if you get a rupture of the blood vessel, if you get a hemorrhage of one of the arteries in the brain? The blood flows freely, no problem. Only slightly more resistance than if it was in the outside air. Zero pressure, right? 0 millimeter pressure. Before the medulla goes through the foramen magnum, there is a structure that has been removed from here. It's part of the dura mater that goes right through here. Would you say that that structure in there kind of looks like a tent? A pup tent? A little like a pup tent? You can imagine the dura mater in there a little bit like a pup tent, right? Well, that's called the tentorium cerebellum. It also has a small opening that the midbrain cannot fit through. Well, the midbrain can fit through it, but what happens is these parts of the temporal lobes here and the hypothalamus, which is just above the midbrain, cannot fit through the tentorium, and they squeeze the midbrain. What happens then? That's called a uncal hernia or a tentorial hernia. It compresses the brainstem. This back here in the tegmentum, this structure here, there's something there called the reticular formation. We're going to be studying that the week after spring break. The reticular activating system. When that is damaged, it is involved in sleep-wake. It arouses the brain by the release of cholinergic acetylcholine throughout the brain. It goes throughout the brain, up and down, and it wakes the brain up. If it's damaged and the brain doesn't have that acetylcholine release, it goes into sleep-like oscillations, which we call a coma. So if you damage this, you produce irreversible coma. Slight damage, maybe it's not irreversible. Take out most of it, it's irreversible coma. So in a tentorial hernia, if you have a higher intracranial pressure, the midbrain and the reticular activating system is compressed by the temporal lobes and the hypothalamus trying to go through that little hole as the pressure pushes it outward towards the foramen magnum, and it causes coma, which is, basically, if you have irreversible coma, you're considered brain dead. Another point on that, a lot of people ask, well, why are the pupils dilated? Why is that a sign of cerebral hemorrhage going really bad? It's because the ocular motor nerves are coming out of the brainstem. And so if you impinge on them and cut their activity to the eyes become fixed. They can't move anymore. And if you withdraw the autonomic innervation to the pupils, they go to their relaxed state, which is open. The third and fourth oculomotor nuclei, which go to the extrinsic muscles of the eye, are right next to the reticular activating system. So when they're compressed, and you're going into coma. We're on to the last structure of the brainstem. Last two structures. The first one is substantia nigra. Have you heard of substantia nigra before, Leslie? Mm-hmm. Hannah? It's involved in Parkinson's disease. The nigrostriatal pathway, and we'll do the striatum later, starts right here. Substantia nigra means black substance. You see any black substance on the brainstem? You see any black substance there on the brainstem? Ladies and gentlemen, that is the substantia nigra, right in front of your very eyes. Now you know where it got its name. Now you know where it's located. It's located in the midbrain, between the corticospinal tract, and the tegmentum, which has the reticular activating system. It's wedged between them. The other structure that is not visible is the raphe nuclei. The raphe nuclei run all the way down the midline of the brainstem. I'm going to put my probe in the middle of the medullary raphe nuclei. There. I'm in the middle of the raphe nuclei right now. But they go all the way up and down the brainstem, not as a pure, even sheet, but they have clumps. And each of the clumps have a different name. One is called the median raphe nuclei, one is called the dorsolateral raphe nuclei. Well, what does raphe mean? Raphe means, if you smash two things together, it's the space that was between them. So it's the two halves, the left and right half of the brainstem smashed together, and that's where the raphe nuclei are. We have white matter, we have gray matter. White matter is white because long fibers have got a substance around them called myelin. Myelin is a type of a fat, it's a lipid. And what it does is it increases the conduction speed of the impulses. I mean, it's like hyperspeed compared to what it would be if the fibers are not myelinated. Unmyelinated fibers go very slow. How slow? Oh, as slow as a meter per second or a centimeter per second. Very slow. Myelinated fibers, they max out at about 120 meters per second, which is, it's moving along at a pretty good clip. The thing is that whether it's myelinated or unmyelinated, there's another thing that determines conduction speed, and that is the size. Larger fibers are faster than smaller fibers. So, of course, what do you want to have in the brain? So you have maximum speed cognition, maximum rapid sensation, maximum motor response. You only want to have huge, honking, myelinated fibers. How big? Well, the bigger the better. This big, right? Won't fit in the cranium, will it? So there's a trade-off. You use myelinated fibers only when you have to. You use them when you have to propagate to a far structure, such as from the precentral gyrus down to the spinal cord. That's a meter away. Then you have myelinated fibers and big ones. So whenever you see big fibers or you see white matter, you're looking at speed reaction time issues. What kind of things are travel very slow? If you remember scalding your hand in hot water ever, scalding your hand in hot water, if you scald your hand, you might remember you get two sensations. The first is the feel of something hitting your hand, the drops of water, without really a temperature sensation yet. And then a fraction of a second later, you notice the thing is hot, really hot. And now the temperature is hit. At that point, you withdraw your hand, either voluntarily or reflexively, automatically. And now the ugly sensation comes in, this dull, aching pain that won't go away. You put cold water on your hand feels cold, it's not cold, but it still aches and burns like crazy, and it'll be that way for minutes. This is all due to fiber speed. The touch fibers are the fastest. So first you feel touch. The alpha fibers are the fastest, that's the touch. The beta is next fastest, that's where you get that temperature sensation. Then you get the gamma fibers. Oh, those are slow. Those are the ones that run like a centimeter a second. Long after your hand's out and everything, they're just still coming in to your brain. And they bring out that achy pain that is just so terrible. And doesn't go away because, they're taking turns and some are faster than others. So there is an example of touch has a priority over temperature, has a priority over pain. Because it's devoting large fibers to it. Same thing is throughout the brain. I don't care whether you're talking about the parts that are involved in intelligence. There are large fibers and there are small fibers. And the small ones are reserved for very local things where speed doesn't matter, where you only have to go a fraction of a millimeter, but the large ones are when you need a quick response that has to go to another area of the brain. The first thing I want you to notice on the cerebellum are these two structures right here. Those are called the cerebellar peduncles. What do you think the cerebellar peduncles attach to on the brainstem? Well, here they are on the brainstem. What are they attached to? The pontine enlargement. The pontine enlargement is a tract that comes from the motor cortex, that's precentral gyrus area 4, problem in area 4, down into the cerebral peduncles, into the pontine enlargement, but doesn't continue to the spinal cord. It terminates on cells in the pontine nuclei that go massive tract, biggest one in the brain, to the cerebellum, to the cerebellar peduncles. Here. The cerebellum then has nuclei in here that project up through the brainstem and eventually reach the basal ganglia, also known as the corpus striatum. Same thing, basal ganglion, striatum, corpus striatum, and are involved in motor control. Particularly the type of motor control where you're learning something new, like to play a piano or play the violin or learning to drive, and at first it's all klutzy and clumsy, and you can't figure out what to do. After a while it becomes automatic, like walking. You don't even think about it. All these complex sequences of muscle contractions happens automatically. That's what the cerebellum is. You could call that motor learning. If the cerebellum is damaged, a person becomes severely uncoordinated, a lot of times with vestibular difficulties, and pretty much as if you are, say, inebriated with alcohol, sort of staggering, jerky, reach for something, whoops, I missed it, that sort of stuff. That's the cerebellar syndrome. Seth said, isn't it amazing how leaf-like it is? Mm-hmm. These are called folia. On the cerebral cortex, we have gyri and sulci. In cerebellum, we just have folia. Folia means flower. It refers to each of these appearing as a flower petal, and you said leaf. So that was almost dead on. The folia of the cerebellum. Beautiful structure, one of the most beautiful. And if you look at it closely, you'll see the folia have gray matter on the outside and white on the inside. So the folia are cortical tissue, specifically cerebellar cortex. That's the cerebellar cortex. Looking here, I will rotate this. Here we have got, my probe is at the junction between the midbrain and the diencephalon. The midbrain and the diencephalon. Okay? The diencephalon, di means two, and cephalon means a bump or enlargement. So encephalic refers to a bump or enlargement. Anything that's a cephalon almost undoubtedly is a cortical structure. Not all the time, but almost all the time it's a cortical structure. The diencephalon is not a cortical structure. It has the same pattern of gray on the inside and white on the outside, but it's one of the last as we ascend upwards. Di means two. The components are the thalamus and the hypothalamus. The hypothalamus is just below this structure. There's a small depression here called the hypothalamic sulcus. You'll have to see it yourself as we pass the brains around. Below it is the hypothalamus and above it is the thalamus. What does the hypothalamus do? Well, you've already seen some of this in our studies already. It is involved in these primitive animalistic biological behaviors. And they include hunger, thirst, sexual drive, anger, rage, love. What are some of the others? Fear. And when you stimulate electrically in the hypothalamus, you can produce all of these emotions in different locations. Also, the pituitary comes out of the hypothalamus. Pituitary is an endocrine regulator, growth hormone, oxytocin for a maternal reproductive system. People call the pituitary the master regulator of the endocrine system. Well, ladies and gentlemen, the hypothalamus is the master regulator of the pituitary. So I think pituitary should be called submaster. Yeah, the pituitary, the stalk of the pituitary goes through a little cranial foramen, and the pituitary is on the other side. So when the brain's removed, you lose it. All you can get is the stalk of the pituitary, which is usually going to be right there, stalk of the pituitary, very bottom part of the hypothalamus, right there. So in addition to these animalistic, biological emotions and behaviors I mentioned, it also all of the endocrine functions, which are huge. The thalamus is a completely different structure. It is in the same thing called the diencephalon. The thalamus really is known as the gateway to the cortex, and it's this thing right here. Thalamus means chamber, and or a foyer, like in a house, a foyer. And it means that it's the foyer to the cerebellar cortex, the house of the cerebellar cortex. It's the entryway. In ontogeny, during development, this thing right here, this bump, this encephalon, which is called the telencephalon, embryologically, dedicated to olfaction, sense of smell. It is the only peripheral nerve tract that runs in the cerebellum. Is the to the cerebrum is this olfactory tract. Look, it goes right into the cerebrum. Olfactory tract doesn't. None of the cranial nerves do. Well, then how do they get to the cerebral hemispheres? Answer is by way of the thalamus. What happened is, ontogenetically, the entire cerebral hemispheres are devoted to olfaction. Huge machinery. I tell you, at that level, olfaction is not what we think of it. It is an absolute analytic chemistry laboratory that can dissect a smell into all its components, remember which components are associated with what, capabilities we don't have. But then, the thalamus develops, and somatosensation, vision, audition goes to the cerebral hemispheres through the thalamus, and they take over that same processing machinery and convert it from olfaction to these other senses that we use all the time. So that means when you're listening to me, understanding me, when you see me, when you hear me, when you grab a brain and you feel the brain, okay, you are using olfactory processing machinery. There was a coup and it was taken over by the other senses, devoted to the other senses. When you're in the thalamus, you're essentially already in the cerebral cortex. You're already there. Why? Because these thalamic structures in the thalamus here, these structures project one-to-one to Brodmann areas in the cerebral cortex. There's a structure right here, where my probe is, called nucleus medius dorsalis of the thalamus, right about here. It projects to one of the Brodmann areas on the frontal cortex. Frontal cortex projects very heavily the gray matter on the surface back to the medialis dorsalis. It's a reciprocal connection. And it's huge. It's massive, and the fibers, most of them are very large in size. So it's for rapid response, right? Rapid response. So as you go different parts of the thalamus, as you go from place to place, you basically move along the cortex. And this is what this diagram shows. And everybody in video land, check this out. Then I'll turn it around so you guys can see. It shows you which parts of the thalamus are reciprocally connected with which parts of the cortex. When you're in the thalamus, you're essentially already in the cortex because the interconnections are so extreme. Ladies and gentlemen, cerebral spinal fluid surrounds the brain. Cerebral fluid is within the brain. And when it's inside the brain, we call it a ventricle. Here you see a ventricle, this hollow area filled with cerebral spinal fluid. Cerebral spinal fluid is in fact made in the ventricles by something called the choroid plexus. You see that grape-like structure there? You guys are going to have to look at this. Go ahead and see if you can find it on here in the base. They look like little grapes. That's where CSF is made. The blood is filtered, turns into CSF. The ventricles go around into the temporal lobe, go through the cerebral aqueduct here, and expand out into the third ventricle, which is much larger. The third ventricle is over the back of the brainstem. This is the floor of the fourth ventricle right here that I'm touching, filled with cerebral spinal fluid. In between the two cerebral peduncles is the fourth ventricle. These are the ventricles. Here we have the giant lateral ventricles, first and the second ventricle. Notice how they come down into the temporal lobes of the brain, which are down, temporal lobe is down in here. There's a little tail off here. The one in the middle here is called the third ventricle, sort of shaped like a quarter. This part here is between the left and right hypothalamus. This one here is between the left and right thalamus. So the thalami and hypothalami are split. When I'm touching the thalamus here, I'm touching the wall of the third ventricle. When I touch the hypothalamus here, I'm touching the wall of the third ventricle. The knife went through, it cut this, it cut this, it cut this, nothing to cut down here. You're going through a cavity. Then down here at the bottom we have the fourth ventricle. It's thin here. The cerebral aqueduct here is the level of the midbrain. The fourth ventricle here is the level of the pons and the medulla. And then the CSF comes out from inside the brain to go around the outside of the brain at holes back here on the sides of the fourth ventricle. If you have a spherical glob of gray matter inside of the brain, such as this, or this, or this, that's called a nucleus. It's just where the gray is more than the white. White more than the gray, white matter is always tracks. It's just the axons of neurons is what a track do. Bundles of axons. Gray matter are the cell bodies and dendrites. Dendrites are not myelinated, cell bodies are not myelinated. Only the axons. So gray matter is where the action is. Tracks are nothing but a communication line to take the signal from here to there. Gray matter is where the processing is done. That's the stuff of the brain. All these very tangled dendrites and cell bodies, it's axons that only go a fraction of a millimeter, all working. That's where we get cognition. That's where we get emotion. That's where we get motor control. That's where we get integration information from our senses to put a whole world picture together of what's out there and where we are in relationship to the world. What puts that whole world picture together? Each area of Brodmann has a piece of it. First, the visual areas of Brodmann, the auditory areas of Brodmann, somatosensory. Then they project to other areas of Brodmann. This one integrates somatic and vision, this somatic and then it goes to another area of Brodmann, and the areas of Brodmann keep integrating more and more. Then you get the motor aspects of the area of Brodmann that integrate the sensory world, what's going on, where you are in it, and then if you move, how you move in relationship to it. And that tends to occur in the parietal and frontal lobes, where you get that really true perception of you in the world interacting with things. And that's what the neocortex does. And it's done. There's an area of Brodmann that is the master one in the frontal lobe that takes care of that recognition. It's sort of where the soul is. One of the things about the brain, you know, if you have a joint here and you want to contract it by this way, the muscle has to go from here to here. And the muscles that go from here to here, here to here, are all similar in function. And things that are close together tend to be similar in function throughout the whole body. The heart muscles, from some lobes of the heart, next to other lobes of the heart, they're all there for contracting the heart and producing the flow of blood in liters per minute. Your fingers all grasp things. This one is related to this one, related to this one. They all work together. In the brain, that's not the case at all. There's no need for it to. Nuclei don't have to be next to each other to interact with each other. They can be on totally different parts of the brain. All you have to do is send fibers there. Send fibers here to here. Doesn't matter where it's located. And so you see that. The brain just becomes scrambled all over the place because it doesn't matter where nuclei are. You just run fibers to it. So because of that, the brain has this crazy layout that makes it so hard to understand the neuroanatomy. And the reason is that. To make matters worse, you have nuclei sometimes, and you have fibers that will go right through the nuclei. So if that nuclei is taken out, you may have a deficit caused by the loss of those fibers of passage. It have nothing to do with the nucleus. And that's one of the big problems in the brain, is he's trying to say, hey, that part is damaged and the person's doing this. You know, maybe that helps people to quit doing this. Well, it could be from the fibers of passage that go through the nuclei, have nothing to do with the nucleus at all. When you especially get to the cortical levels, you get this holistic thing going, where, especially in the frontal and parietal lobes, how much of a deficit you have depends on how much brain you remove. And it doesn't really matter where you remove it. Because the brain sort of functions holistically at that level, in certain parts of it. So you can't say what is the function of the brain here, because its function depends so much on interaction with all the other lobes that that doesn't make any sense anymore. So you truly get into that holistic thing where the sum of the parts is greater than the whole. But here we have the temporal lobe, occipital lobe, parietal lobe, frontal lobe. Here we have superior temporal gyrus, middle temporal gyrus, inferior temporal gyrus. Here we have superior frontal gyrus, middle frontal gyrus, inferior frontal gyrus. These are the orbital gyri, very important in sight, guys, the orbital gyri, as are the frontal cortex. Do you see that this forms a depression here? That's because it's right over the globe of the eye. The eye is right here. And because the eye is spherical, it forms this sort of spherical type depression here. It's called orbit because it's the orbit of the eye, and so is the orbital gyri. If we come around here, here's the central sulcus. We have the precentral gyrus, the postcentral gyrus, the parietal lobule. The parietal gyri in sulci are notoriously different in every person. So there is no attempt made to identify individual gyri. It's just called a lobule. So this is the parietal lobule. Notice the parietal lobule is above the superior temporal gyrus, behind the postcentral gyrus. Coming to the back, we have the superior, middle, and inferior occipital gyri, which come around here and fuse. This here is called the occipital notch, and it's considered a dividing line between the parietal lobe and the occipital lobe. It's an arbitrary dividing line. It's just an anatomical structure. This is the inferior temporal gyrus here. As you get out to the front part of the temporal gyri up here, this part here is very close to the midbrain. Like I touch here, it's the same as touching here or here. If you all can see that here, here or here, the same as touching there. So it's right next to the brainstem, right next to the midbrain. In fact, there's a protrusion out here. It's not too visible on this particular brain structure. It is here. Wow, it's beautiful here. If everybody can see from there, stand up if you have to. This is the uncus. It's on the inferior temporal gyrus, and it's a little bump called the uncus. Very important. Look how close it is to the midbrain. The uncus compresses the midbrain if the intracranial pressure gets too high. and causes the patient to go into an irreversible coma. That third nerve, the oculomotor nerves, get caught up in that, causing fixed and dilated pupils. I'm going to put my probe into the center of the amygdala. Are you ready? My probe is in the center of the amygdala. It's 1 centimeter below the uncus. This is the cingulate gyrus. The cingulate gyrus. See the septum pellucidum? Cingulate gyrus. This is the precentral gyrus, postcentral gyrus, parietal lobule, cingulate gyrus, septum pellucidum, or the ventricles, thalamus, hypothalamus, thalamus, septum pellucidum. Then we have this baby right here. This is all white matter. Ladies and gentlemen, this is the corpus callosum. Corpus callosum means big body. Colossal. Colossal structure. Corpus callosum has one function only. Connects the left neocortex with the right, with fibers. Fibers that connect left and right like that are called commissural fibers. A commissure. Commissure, anything commissural, means left to right. In the same way that the thalamus has a one-to-one relationship to different parts of the cortex, commissural fibers do too. This part of the cortex has strong connections with this part of the cortex by fibers that go down through the corpus callosum and back up. This part here has strong connections to this part here by fibers that go through the corpus callosum and here. Occipital to occipital, orbital to orbital, through the corpus callosum. Very important for left-right coordination. The left and right halves of the brain are very coordinated until you cut the corpus callosum, which is done surgically to treat certain types of epilepsy to keep epilepsy from spreading to the other hemisphere. When this is done, people with that section appear every bit normal in everyday life. But if you do particular tests on them, you find out that there's 2 people living in one. Roger Sperry got the Nobel Prize for discovering this. There are 2 people living in one body. Person on the left, person on the right. And they have different capabilities. The one on the left is better at calculating, speaking, linguistics, spatial orientation, solving puzzles. The one on the right is better at intuition, sensing when things are right, when things are wrong, and so forth. Normally they work so closely together, there's no difference at all. It's only detected there, and that's where you get this idea of the, are you left brain or right brain? Well, everybody is both, unless you split the corpus callosum. This bump of gyri starts to go forward a little bit, then backward, and around to the side. So we have structures here that, before it did the ram's horn thing, that were spherical. After the ram's horn, they were dragged down. So they're long now. Not only long, but they form an arch. And you can see it in the ventricles. The moved forward here, then the ram's horn around to the side. The ram's horn thing's really important. 3D, always remember ram's horn, always remember how this looks. So we have these structures that are dragged around into the temporal lobe, and they form this ram's horn type pattern. Okay, here we go. Everybody on the video land, this is the corpus callosum. Does it look familiar to you? Corpus callosum. So you know the third ventricle is down here, and the hypothalamus and thalamus are down here. These are the olfactory tracts coming in. Notice how this structure is dragged all the way down into the base, into the depths of the temporal lobe. This structure here is the amygdala. You go all the way to the temporal lobe, go to the medial side next to the brainstem, there's a bump. There's a little bump on there. That's the uncus. The amygdala is down there. So the uncus is right on the inside of it right here. That's where the uncus is. the uncus of the inferior temporal gyrus. That's where the amygdala is. It's still spherical. This structure right here is the hippocampus. Hippocampus, the big end of the hippocampus is where the amygdala is located. It gets smaller and smaller and smaller and smaller and smaller, ends somewhere around here. Amygdala and hippocampus work closely together. They are among the most primordial structures in the brain. They develop to their full extent long before the cerebral cortex starts to develop. And they stay pretty much the size that they are while the cortex expands, 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 as the fetus develops. Very important in psych. Hippocampus, think spatial orientation and memory. Alzheimer's, hippocampus is one of the first structures to get inundated with the amyloid plaques, and memory is the first thing to go. Amygdala is involved in emotion, very closely connected with the hypothalamus. Animalistic emotions, especially fear. Amygdala and fear are very closely related. You can damage the amygdala and make a person courageous by damaging it. Or if there is a stroke that affects the amygdala, a person can become courageous, fearless. No anxiety, it's a cure for anxiety. Don't need any benzodiazepines. Just take your probe, get into the amygdala. Everything's good. Another important structure that is important is a tract that goes from the hippocampus to the mammillary bodies. The mammillary bodies are located in the base of the hypothalamus. Here's the left mammillary body. That round thing is the left mammillary body. The word mammillary body comes from female mammillary glands, and if you look into those, you see that there's a resemblance. So a very colorful name by our ancestors. The mammillary glands receive a projection from the hypothalamus. Well, hypothalamus is down here in the temporal lobes, and it's there. How does it get there? The answer is by way of the fornix. It goes from the hippocampus to the mammillary bodies. Why is it called the fornix? It comes from the same word as fornication. A forn or fornix means C-shaped. Its function is not known, but it is part of the limbic system, and anything in the limbic system is important in psych. If you look at a cross-section of the white matter, of the cortex, you will see gray matter all along the edge of the gyri, right? There's gray matter above white matter. And if you look at it, there is so much white matter. Tons of white matter underneath. This thin sheet of gray and this bulk of white matter that makes up the bulk of the cerebral hemispheres. But there are some places where you see something different. It looks wrong. You see gray matter down in the white matter. Down in the white matter of the cerebral hemispheres. That's known as subcortical gray matter. What is it doing there? There are 3 subcortical gray matters. First is the hippocampus. Hippocampus is embedded down in the white matter of the parietal and especially the temporal lobes. And the amygdala is subcortical gray matter. There's gray matter, white, and all of a sudden there's a gray matter nucleus embedded in the white matter. In the tip of the temporal lobe, that is the amygdala. And there's one more. It's the basal ganglia, also known as the corpus striatum. It is embedded in the white matter. Where? If you go to the wall of the lateral ventricle, I push my probe in, I'm in the gray matter of the basal ganglia, the lateral wall of the lateral ventricle. In other words, here's the lateral ventricle, basal ganglia is right here. Right here. Basal ganglia are located right here and right here. They have three major components: the globus pallidus, the putamen, and the caudate nucleus. The three are essentially function as one body. They have white matter that separates them. Remember what I said, it doesn't matter where you put the nuclei? They have so many connections that they really function as a whole. Now, the globus pallidus and the putamen are sort of egg-shaped, way deep right in the center. Cerebral hemisphere is down here. The caudate nucleus is dragged down into the temporal lobes. Caudate, a caudus is a tail of an animal. And the caudate nucleus looks like a comet with a long tail. And that's where it gets its name. Caudate means tail-like. The hippocampus is largest in the temporal lobe and thins out as you go to the center of the cerebrum. The caudate nucleus is biggest in the cerebrum and gets thinner as you go down. And they form an interesting, it's like yin-yang. You know how yin-yang is one gets smaller, the other gets bigger? The caudate nucleus and the hippocampus do a yin-yang thing as they go through. Caudate gets smaller, hippocampus gets bigger. Have you visualized that? Okay. We have the concept of subcortical gray matter. They are hippocampus, amygdala, and basal ganglia. Basal ganglia has three components: globus pallidus, putamen, and caudate nucleus that work together. Now we're going to finish up the day by looking at the plastic embedded sections. We're going to go inside the brain. I see gray matter right here, deep within the white matter. Ladies and gentlemen, this is the striatum, the basal ganglia. Look down in the depths of the white matter, there's a gray structure here. Yes, that's the basal ganglia. That's the striatum. You can't see stripes at this level because it's a little too lateral. But what we're cutting through here is the putamen itself. Here's an interesting one. We see the substantia nigra of the midbrain here and the thalamus. The substantia nigra is projecting to the basal ganglia, which is lateral to this thing. Here's the corpus callosum. But there's the substantia nigra. The basal ganglia are best seen, everything is best seen on a frontal section. Now you have to visualize, guys, the C-shaped thing. Because of the C-shaped thing, that means that in a certain section, you're going to see hippocampus up here and hippocampus down here, right? You're going to see caudate nucleus up here and caudate nucleus down here. Keep the 3D anatomy in mind. Out here, we have got the basal ganglia. This is the putamen, a small structure called the extreme capsule, which is neither here nor there. And on the walls, remember how I pointed through? I said, I'm pouring through the walls of the lateral ventricle, and I'm hitting the caudate nucleus, one of the components of the basal ganglia. If you look right here, you will see some gray matter on the wall of the lateral ventricles, here and here. So when this comes around, you pay attention. Caudate nucleus, and then the putamen and the globus pallidus. The fact they're separated by this tract, which actually is the corticospinal fibers, going down into the spinal cord, it's just cutting through the basal ganglia. Not very many connections with it. In the frontal sections, you not only see the caudate nucleus and the putamen there. Here again we have, when this comes around, caudate nucleus, basal ganglia, all lateral to the lateral ventricles. The caudate nucleus and the basal ganglia form the walls of the lateral ventricle. And there they are. Let's talk hippocampus. Subcortical gray matter, medial part in the white matter of the temporal lobes. Okay, Alex? I'm going to point to something. We have the gray matter, temporal lobe, and we have the white matter. Then you look at subcortical gray matter, but the hippocampus is itself a cortical structure. So you'll see this stripe of gray matter, and it's convoluted, like that. Hippocampus is the Greek word for seahorse. And the hippocampus in a cross-section looks like a seahorse. It goes like this. Like that. It is. See, the hippocampus goes forward and forward in the temporal lobe, and then there's the amygdala at the end. It doesn't become the amygdala, but the amygdala is at the tip of the hippocampus. We're going to close this out. I have a little lecture before we go. We're going to go over and find an amygdala on the brain specimens. You know, we have these conceptions, things that are presented in pharmacology textbooks, such as the substantia nigra pathway, releasing dopamine, or having a reduction of dopamine. The dopamine causes the tremors and the akinesia, you know, the stiff body, inability to move, loss of will movement in Parkinson's, and the shuffling gait of Parkinson's. And they almost talk about it like, to me, it's sort of like a hose or something. You know, here's the dopamine and there's a hose sprinkling into the substantia nigra. Very crude, sort of a hydraulic almost analogy. Let me tell you something. Dopamine is found in every structure of the brain. Norepinephrine. Serotonin is found in every part of the brain. Acetylcholine. Do you guys see this little piece of brain here? Can you see it? I don't know where that came from, but it has got all 57 known neurotransmitters and neuromodulators inside of it doing stuff, plus another 100 that we haven't discovered yet. Every part of the brain uses all the transmitters. So you say, okay, well, what happens there? You know, if we jack up norepinephrine in a person, jack it up, they become energetic and happy. It's used to treat depression. What's going on there? Well, I'll tell you what, if you cut the spinal cord, and in the isolated spinal cord, which contains just everything from the navel down, just lower half of the body, basically, innervation there, and you soak that thing in norepinephrine, you know what happens? The legs start to walk. The norepinephrine turns on the locomotion pattern generators that control the sequence of movements to the muscles to produce coordinated forward locomotion. Well, wait a minute, is norepinephrine an antidepressant? Is it a happy thing? Or is it locomotion? It's both. If you keep jacking up the norepinephrine, long after the person becomes totally happy and energetic, they'll start to have involuntary leg movements as the locomotion pattern generators are turned on. And perhaps in the hippocampus, let's say there's something in there that where norepinephrine is involved in, I'm making this up, involved with memories of our childhood. You'll be happy, have memories of your childhood, and involuntary locomotory movements. If you jack it up more, pretty soon you'll bring in all the brain functions, which will be completely disabled at that point. So when we give drugs, really, and you say, well, what does it do in the brain? It's really the first thing that's the most sensitive that is produced. Because there's thousands and thousands of things that transmitter is involved in. Just is the first thing that's produced. Ideally, we could deliver it locally to different parts of the brain, and then the same neurochemical, the same neurotransmitter, the same pharmacological agent could produce cures and treatments for all kinds of things. But we don't do that yet. I don't know if that day will ever come. It's fairly invasive. But when you remember, when you're doing pharmacology, all transmitters are involved in everything. It's just a matter of which one is the most sensitive when you're given these pharmacological agents. And I think that is it.